We will have a brief introduction on bioelectronics. So what are bioelectronics? Bioelectronics is basically the integration of your biomolecules with electronic elements to yield functional devices. So one example of that is your brain-computer interface, whereas a certain electrode can be embedded on the surface of the brain in order to detect the electrical signals from there. Another one is the chips embedded inside your subcutaneously, so just beneath the skin, or even neurons growing on a circuit board. So another functional example is as part of the DNA sequencing devices. So you have DNA hybridized to a chip, and that's a sensor. So the sensor senses the presence of the DNA that was attached to your chip. So those are some examples of bioelectronic devices. Now, why do we need to include biological systems in our electronic device? Why not just use simply electronic devices? So, for one thing, biological systems produce very effective catalytic systems. In fact, they are mostly used in the industry. So, when we say catalytic systems, so these are they speed up the reaction. So, catalysts are elements that speeds up the reaction without consuming them. And for biological systems, the catalysts that we are talking about are called the enzymes. These are specialized proteins that speeds up a certain reaction, and they are very important in the not only in research but also in the industry. For example, we have invertases. They break down sucrose to form the glucose and the fructose. So in effect, you can produce a fructose component from that. So without invertase, it's very difficult to do. So it will require very harsh conditions such as high temperature or high pressure in order to do that. Whereas we can do with enzymes, you can just do that under mild conditions. Usually, just a bit warm, so around body temperature types. So those are examples of catalytic systems in biological, catalytic systems in bio or biological organisms. Another one is their high selectivity and specificity. So enzymes again have very high effectivity and selectivity. Actually, not only enzymes, even the antibodies. In today's pandemic, if you know the high speed testing that involves antibody testing, so those makes use of the high specificity and high selectivity of biological systems. In particular, they're the antibody itself. So they can recognize very specific molecules that it will take a while for us to design a non-biological system for that. So we just harness the biological system and input it in our electronic or biosensor system. So another one is their fast and complex computing and imaging system. Case in point, you have the brain. So the brain, there are many analogies for the brain as a computer. So actually our brain has a very high memory capacity, higher than any computers to date. Now you have your, of course, when we visualize, for example, when we see things, the light is sensed by our eyes and then transferred to optic nerve, and then you have your brain, or rather the optic nerve passes it to the visual cortices, and then the cortices will help integrate the two structures, and then your brain will analyze what am I seeing. So if we make as of now, the technology now of making an imaging system or an imaging software is much more what do you call that, much more advanced compared to several years ago, but still it has a certain lag time, whereas when we do that, as soon as we just do that instinctively, and it's quite fast now without much complications. So another one is again in the brain. For example, for the brain, you have processing and storage of three-dimensional information. So the biological systems have compartmentalizations. So they compartmentalize several parts. For example, in thinking or in several processes in the brain. We have areas that function for language analysis, logic, emotions, etc. So those are compartmentalization processes. So in order to take advantage of those qualities of biological systems, we usually integrate them into electronic devices. So you have the biological system and then integrate it with electronic devices in order to create a bioelectronic system. And how do we do that? So this is the basic configuration of your bioelectronic devices. Actually, there are two. So the first one is that you have a biological event. For example, it can be in a soup or in a mixture of solution, whereas you have lots of things in their proteins, mixture of different components, and then you have your antibodies, or you are taking advantage of the selectivities of the biological system. For example, antibody. For example, let me give you a more concrete example. The how do you sense antibody testing? For example, again, during the in light of the COVID crisis. So how does the fast, how do they know that you were infected? That the fast antibody testing. So for example, you take the blood sample. So the blood is a mixture of different components. So you have the blood cells, the different nutrients that your body has, different waste products, as well as if there is 1 in a million chance, well, not exactly not 1 in a million, but a very even a very very small amount of protein, of the viral protein in there, the testing kit, which contains antibodies, can bind to that protein and no other. So they're quite selective. They immediately bind to that protein, that is like the protein of the virus. And then once it binds, you have a transducer that sends the binding. So it can be it can produce an electrical signal, or there can be a change in the mass. Usually it's change in the mass of piezoelectric. So you have that sensing. So you have a signal, and then that is integrated into your computer and read as, oh, it's positive, or if there isn't any, so, oh, it's negative. So something like that. So that's the integration. So you have a biological event happening first, and then you have your electronic devices sensing that event. Now the second configuration is the other way around. So in this case, you have your electronic unit that sends signal to a biological system. So example here is a proposed mechanism of allowing the blind to see. So you have a camera mounted on a glass, and then you're just basically simulating how the eye works. So you have your camera that sends a signal to your computer that creates a signal that the brain can understand. So you have the brain, or rather an implant on your brain that can send the signal directly to the brain, so allowing to see. So that's one example of an implant. So that's the other way, the other configuration. Although this configuration is rarer than the first configuration, but there is also this possibility of a configuration. So in bioelectronics, there are many possibilities that you can do. So you have these are certain different fields of interest in bioelectronics. So you can do biosensors. It's actually one of the most largest field there. So in biosensors, you can do biorecognition or biocatalytic systems, or in creating biofuel cells. So you are actually taking advantage of chemical reactions that are normally happening in biological systems, and then generating electricity through them. So this is a biofuel. If I remember, Mapua had a project to developing biofuels a few years ago. I'm not sure if it's still continuing, but anyway. So another one is a neuronal network interface with semiconductor microstructure. So creating neuronal networks. Actually, did you know that neuronal networks are modeled after how the brain works? So in neuronal networks, you have processes or computers linked together in the same manner. That is actually how your brain functions. You have different neurons that are interconnected or linked together. So together, they process information in order to create, basically our intelligence is basically how that happens. And then in the field of nanotechnology as well. So very, very small scale sized of systems. So basic components of bioelectronic devices, you have electronic elements or support, of course. This is for the electronic side. So they can be electrodes, field effect transistor devices, piezoelectric crystals, magnetoresistant recording medias, or even the tips of the scanning tunneling microscopy, or even AFM, atomic force microscopy. And then, of course, for it to become a bioelectronic device, you need a biological material or a biological element that is integrated or linked to your electronic support. So, even though it's a fundamental requirement, or rather, the fundamental requirement of any bioelectronic system is, of course, since you have two different systems, the electronic and the bio, you need to couple them. So, the electronic coupling is very integral between biomolecules and electronic support. They must be able to communicate with one another. So, some examples of how does coupling is attained. So, for example, this is a biofuel cell. So you have a certain reaction happening in the anode. So it can be sometimes it's actually a cell, a biofuel cell that involves microorganisms that perform redox reaction on substrates. So when they do redox reactions, if that cell or even an enzyme is linked to a mediator support, so when it catalyzes a certain reaction that actually involves transfer of electrons. The electron is transferred to the mediator and then transferred to the anode, and then if that anode is connected to a circuit, so you will generate actual electricity. And then the same goes for the cathode, that's the electron acceptor. So you have completed the circuit here, and you can generate electricity by just doing enzymatic reactions or even growing microorganisms, the redox microorganisms in a certain anode and cathode, so you can actually generate electricity. So another example is an enzyme that is embedded in a certain polymeric film. So for example, here's a polymeric film. The R's there are polymeric film. So these are polymeric film, these are enzymes that catalyze a certain reaction, and this is usually used for sensing. So when it catalyzes a reaction, there's an electron. That's actually how you one of the ways on how you couple your biological system in an electronic device. So it's the most basic one is of course electron transfer. So you have electrons moving, ending up in the electrode, which can be sensed because it's basically a current output. So you have this type of sensor involving immobilized enzyme. Okay, so even though it's a fundamental requirement, it's also a fundamental difficulty. So it's integral to couple them, the bio component and the electronic component, but it's also very difficult. So the lack of electrical communication between biomaterials and electronic elements presents one of the fundamental difficulties in designing bioelectronic systems. So that's it for this video. In the next video, we will be discussing about biosensors.